Thank you very much. Today I've been asked to talk about the role of innate immunity in the pathogenesis of serious non-AIDS events. I'll start by talking about how HIV and inflammation are linked and how breaches in the gut mucosa trigger the production of pro-inflammatory cytokines and can predict the development of serious non-AIDS events, how monocyte activation impacts on innate immune pathways, and I'll mention links between monocyte metabolism and inflammation. Lastly, I'll just briefly touch on innate immune senescence and the risk of serious non-AIDS events. But it's important to note that these lines are just dotted and that there is a lot that we don't know at this stage, in fact, huge gaps about how these pathways interact and loop back on each other. Chronic immune activation and inflammation are both central to HIV pathogenesis in both treated and untreated individuals. This actually starts during seroconversion, when there's an intense cytokine storm with elevated plasma levels of a number of cytokines, including pro-inflammatory cytokines such as TNF. This is accompanied by intense T-cell activation, and T-cell immune activation declines over the period of time when suppressive ART is provided. However, it's important to note that even though there's this dramatic decline in T-cell activation, shown here with CD8 positive T-cells, we find that the levels never really reduce to the same level that we see in uninfected individuals. So that's a little bit about T-cells, but I'm going to talk to you today about monocyte activation, innate immunity, and pro-inflammatory pathways. Let's start by just asking what we mean by pro-inflammatory pathways. That's when you activate cells such as monocytes and macrophages that produce cytokines, including IL-6 and TNF-alpha, that can result in an inflammatory environment. Uncontrolled chronic inflammation leads to fibrosis and end-organ disease. Now, the roles of T cells and monocytes differ at varying stages of HIV infection in some ways. T cell activation has its main pathogenetic effect in untreated individuals, where it's tightly linked with CD4 cell death and T cell decline. Monocyte activation, on the other hand, is perhaps now being found to be more important in the pathogenesis of the longer-term non-AIDS morbidities that are particularly involved in chronic inflammation. And CD16 positive pro-inflammatory monocytes are increased in the setting of HIV infection and only partly normalised by combination ART. If we talk about monocytes, they're really divided into a number of subsets. And for this purpose, I'd just like to talk about CD16 positive monocytes, which can be further subdivided into non-classical and intermediate monocyte subsets. These CD16 positive monocytes are generally considered to be pro-inflammatory. They're a minor subset in the blood compared to the classical monocytes that are more commonly seen. So these CD16 positive monocyte subsets are increased in patients with uncontrolled viremia and may normalise with ART. This study here from the Mike Lederman lab shows that in orange, it shows that CART restores the proportions of monocytes in blood. However, patient populations differ. And for example, CD16 positive monocytes remain elevated in elite controllers. We also find from data from our lab that pro-inflammatory CD16 positive monocytes can remain expanded despite ART and virologic suppression. If we look at monocytes from HIV-infected patients, shown here in blue and red, we see that there is an increase in intracellular pro-inflammatory cytokine production that is present for IL-6 and TNF in all monocyte subsets, compared to what's shown in black, HIV monocytes from uninfected individuals, where there are lower levels of these pro-inflammatory cytokines. Now, some really strong evidence has come just recently from the Landay lab, where they show that non-AIDS events can be predicted by markers of inflammation and coagulation. This was a case control study in virologically suppressed patients where blood was taken for inflammatory markers at baseline, 1 year post-ART, and at pre-event, and then subjected to conditional logistic regression analysis. What this study found was that if you look at the markers of inflammation and coagulation, shown here in the red box, these were able to predict the occurrence of serious non-AIDS events at baseline and also during ART and pre-event, whereas the CD38 positive CD8 activated T cells were not associated with that. Thus, increased innate immune inflammatory markers seem to be strongly associated with the development of non-AIDS morbidities, whereas our current evidence would suggest T cell activation may be less related to these conditions. Where does inflammation come from? You're going to hear a little bit in the next talk from Peter about HIV and co-pathogens, but so what I want to talk to you about is that there is a profound loss of CD4 positive T cells from the gut-associated lymphoid tissue. This starts in early HIV infection and results in a permanently damaged gut mucosa. It allows the translocation of LPS and other microbial products across this leaky gut and results in systemic inflammation. All this is incompletely restored by ART. In fact, there's a lot of residual damage to the gut that persists through the course of HIV infection. Meanwhile, macrophages accumulate in the damaged gut mucosa, particularly in untreated individuals, and they can further drive pro-inflammatory cytokine production. As a consequence of increased gut permeability, gut-derived bacterial products are able to move from the gut through the liver into the systemic inflammation, into the systemic circulation, where they result in or contribute to an environment that is of inflammation and immune activation. And this contributes, we think, to end organ disease. But it's not as simple as all this. Translocated LPS can also activate the Kynurenine IDO pathway. That's usually responsible for converting tryptophan into the neurotoxic quinolinic acid. But activation of this IDO pathway can also result in innate immune activation and pro-inflammatory cytokine production. And to add to this, there's now a lot of evidence that an altered gut microbiome, an altered gut microbiome, meaning a change in the composition of the gut organisms, can also activate the IDO pathway and contribute to pro-inflammatory cytokine production. A paper that's just out by Peter Hunt's group shows that this IDO pathway can also be linked with premature mortality and with incomplete CD4 T cell restoration. You can see here that there's a higher kynurenine to tryptophan ratio shown in blue in patients who have a higher mortality, and that's shown at 2 years after ART initiation. The gut-derived bacterial products can also go to the liver and stimulate Kupffer cells and hepatic stellate cells. We know less about this, about the pathogenesis here, but it seems that both of these can then in turn activate innate immune pathways, pro-inflammatory pathways, and pro-fibrotic pathways, contributing to liver damage, decreasing the clearance of LPS, which then exacerbates this whole loop and contributes to the development of end organ disease, including liver fibrosis. This, of course, is then further complicated when you have co-infection with hepatotoxic viruses such as hepatitis C, which can then worsen the results. Recently, a study that was performed in Uganda showed that monocyte activation is associated with liver fibrosis in infected patients in Uganda. HIV-infected and uninfected subjects with liver fibrosis were studied. There was less than 5% of patients who were co-infected with hepatitis B, and I think none were co-infected with hepatitis C. What they found overall was that monocyte activation was a good marker of liver fibrosis. So patients with monocyte activation had higher odds of liver fibrosis. This was measured by soluble CD14 levels. There were a risk overall of adjusted risk of 1.19, whereas if you were HIV infected and you also had higher levels of soluble CD14, that further increased the odds of liver fibrosis to 1.54. So how do monocytes contribute to the development of cardiovascular disease in the setting of HIV? Well, we all know that the pathogenesis of cardiovascular disease is multifactorial. However, monocytes and chronic inflammation play a crucial role in the development of cardiovascular disease. Added to this, though, are a number of other pathways, including procoagulant pathways, critically important, but I don't have time to discuss those today. If we look at how HIV contributes to the development of atherosclerosis in the setting of HIV and focus on what it does to monocytes and to endothelial cells, we see that there's an endothelial and monocyte activation with an increase in expression of adhesion molecules on the surface of these cells that increase migration of monocytes into the growing atherosclerotic plaque. There, in the setting of a pro-inflammatory state induced by HIV, these monocytes take up pro-atherogenic lipids, including oxidized LDL. There's also an increased transition or differentiation of these lipid-containing monocytes into foam cells, which is exacerbated by HIV. Just some in vitro data from our lab shows that HIV does promote the formation of monocytes into foam cells in vitro after these cells migrate across an activated endothelium. It was critically important that the endothelium was activated and also that HIV was shown to augment this process. I'd like to just mention how monocyte metabolism can augment inflammation in the setting of HIV. Glucose is needed in the cell for energy production, and activated monocytes dramatically increase their expression of GLUT1 on the surface and concomitantly increase glucose uptake and switch from oxidative to glycolytic metabolism. In the mouse, GLUT1 was shown to be rate limiting as a transporter for glucose into macrophages. This is also true in humans. Macrophages also with high GLUT1 expression have been shown to express high levels of pro-inflammatory cytokines IL-6 and TNF. Some unpublished study from Clovis Palmer at the Burnett and Josh Anzinger from the University of West Indies shows that there's an increase in the proportion of GLUT1 monocytes in HIV infection, shown here, and you can see that it is not restored in patients who are on ART. This correlates with monocyte activation by traditional markers and also with D-dimer expression or production. The increase in GLUT1 expression has been shown to be limited to the intermediate and non-classical monocytes in HIV, shown in red, and not restored by ART, shown in blue. I'd just like to finish by saying, does premature innate immune senescence contribute to the premature risk of non-AIDS morbidities? Well, both ageing and HIV can contribute to result in immunological changes that are associated with chronic immune activation and inflammation, and both you see evidence of immune senescence, although this may be a little difference in the two populations. And these are linked to the risk of all the diseases that we heard in Dominique's presentation. Short telomeres mean the cell has aged, and this is the best evidence that we have for innate immune senescence. And what we see is that monocyte telomere length is shorter in HIV-infected individuals of a young age 29 years here. Very similar monocyte telomere length to what you see in black in young uninfected individuals. And in fact, similar in HIV to what we see in elderly uninfected individuals with a median age of 72. So HIV induces changes to monocytes in men and women, studies we've done with Alan Landay's group. These changes appear approximately 10 to 14 years earlier in HIV-infected compared to uninfected women. But as we've just heard, there's no good evidence that clinical phenotype of ageing emerges earlier in HIV patients who have innate immune senescence. So in summary, the pathogenesis of non-AIDS events is complex, multifactorial, a pro-inflammatory environment associated with coagulation, fibrotic and immune metabolic changes seems to be prevalent and heighten the risk of these non-AIDS events. Most of these immune changes are only partly reversed by ART, resulting in a residual immune dysregulation syndrome with persistent activation of a number of immune pathways. The best evidence we have so far that the innate immune system is involved with non-AIDS events comes from the study showing that circulating biomarkers of inflammation predict non-AIDS events with less evidence for T-cell involvement. And there's no good evidence at this stage that whilst innate immune senescence occurs earlier in HIV, that this is translated to premature risk of serious non-AIDS events. I'd just like to finish by thanking all of my colleagues at the Burnet Institute and our other colleagues who've contributed to this study who are listed here, and I won't mention all the names in interest of time. Thank you.